And today, what I'd like to do is welcome our presenter, Dr. Jonathan Fuchs. Dr. Jonathan Fuchs is a research associate in computational biomedicine at Weill Cornell Medicine and works with Dr. Chris Mason, who gave our epigenome webinar yesterday. Jonathan is a bioinformatician interested in developing pipelines for functional genomic analysis. Like I'm assuming several of you in the audience, he's proficient in Python, R, and Bash, and uses these languages to analyze high-throughput sequence data. Currently, Jonathan is developing reference materials for sequencing reproducibility and epigenetic landscapes of well-characterized cell lines. Jonathan's background is in evolutionary biology, having performed research for his PhD on the phylogenomics of Myxozoa, a group of bizarre, that's his words, not mine, microscopic endoparasites of economically critical fish around the world. Jonathan explored the genomic underpinnings of how Myxozoans, whose closest living relatives are Medusozoans, jellyfish, hydras, cube jellies, became so radically different in biology, ecology, and life history. Jonathan, we're glad to have you this morning. Let's hear from your SEQC2 group and what they've been doing.
Excellent. Thank you, Dr. Jones, for that introduction. I do miss working on those truly bizarre parasites, but I'm excited to share a little bit of our work today. So let me share the screen. And do you need me to hit the record button, or is it already recording? Looks like it might be already.
It is already recording, so please go ahead and share the screen. Thank you.
Excellent. Great. All right, so let's pull this up and go full screen. And all right, can you see my full screen? See me, hear me? Does everything look good?
Yes, looks good. Thank you.
All right, excellent. So thank you again, Dr. Jones, for that introduction. And thank you all for being on this virtual webinar today. It's my pleasure to present alongside many other excellent researchers and excellent work being done and presented in the MEQC webinars and the SEQC2 consortium. We're excited to publish this paper alongside a series of other papers in a joint session between Nature Biotechnology and Cell Genomics in the next couple of months. This morning, I want to share with you one of those studies that I and several other people have been leading over the last couple of years, a multi-platform assessment of DNA sequencing reproducibility. So genomic DNA sequencing across an array of next generation platforms. And we leveraged both human reference materials and some bacterial reference materials in the study that I will break down for you. So a little bit more detail about the background and what our aims were. This is a follow-up to previous work also published in Nature Biotechnology, looking at reproducibility of RNA-seq data. And in this case, we're looking at DNA-seq genome-wide. The idea is to take a set of well-characterized reference samples, which I'll explain in a little bit more detail in a moment. But we take these sort of samples that are used for benchmarking and genomics research development, and we generate a lot of intra and inter-lab biological and technical replicates. And we wanted to model the expected or likely range of performance for each of these sequencing platforms, compare and contrast the relative strengths and weaknesses, and provide a kind of ground truth, might be a bit of a strong term, but something that could be used as a reference set or a benchmark to model against something that a sequencing facility or a core facility might generate with their own NGS machines to enable their own genomics research. So this is intended as a public resource. This is something that we feel very strongly about providing freely. All the data is available in SRA and even on our own S3 endpoints for anyone who wants to collaborate with us directly, as well as all of the code used to generate the analyses, the figures, and everything in between. We want to make it as free and open and reproducible as possible. And the other thing worth noting is that even though we are comparing and contrasting these platforms, this isn't really intended as a bake-off. We're not trying to say that any one platform is per se better than any other or the best. So with apologies to anyone who's a fan of the Great British Bake Off show, this is not going to be that, although it is a pretty good show, and I would recommend it as good, easy watching for anyone who's interested. But anyway, to give a little bit of background, as I mentioned about these samples, there's some really great work being done through a group at the National Institute for Standards and Technology, or NIST, the Genome in a Bottle subgroup, which if you're like me and you've been working with these samples for many years, you kind of lose sight of the fact that this is a clever pun on the play on the phrase genie in a bottle. So got to keep things light and humorous. But Genome in a Bottle, or GIABL, is a dedicated to creating and providing reference materials for authoritative characterization of human and other genomes. So the idea is to take a set of cell lines, this was about 5 years ago, and establish them as these reference benchmark sets. So taking EBV immortalized B lymphoblastoid cell lines split between NIST and the Coriell Institute for Medical Research, the idea is that there would be these large homogenized batches that could be aliquoted and distributed to labs for reproducible research. And not only is Genome in a Bottle developing these as a resource in terms of samples, but also in terms of technology and workflows and files to enable research. Genome in a Bottle has a really great GitHub repository full of bed files, Jupyter notebooks, anything that a bioinformatician might want to enable their own benchmarking work. So I highly recommend anyone on this call who's looking to get into this kind of work to explore this resource. It's really great, and everyone behind it is very open and very willing to help if you have any questions at all. More specifically, the cell lines that I have mentioned, there are 7 of them that are routinely used for characterization that are now known as the NIST reference material A398 or NIST RM A398. There are 7 cell lines in total that are commonly used. If you watched Dr. Christopher Mason's talk yesterday on our study interrogating the epigenome of these samples, we characterized the epigenetics of all 7 cell lines. For this study, we were really just focused on HG002, 3, and 4, the Ashkenazi Jewish family trio that are consented through the HapMap project for reproducible research. And the other cell lines are also, studied in other related works, but in this case, we wanted to focus on those alongside the bacterial references. So diving into the figures themselves, this is kind of an overview of what's the universe of samples and platforms that we explored within this work. So stratified into human and bacterial, on the left, we have the aforementioned HapMap trio, the Ashkenazi Jewish trio, HG004, 3, and 2. And we characterize these across an array of well-established, commercially available, commonly used sequencing technologies, instruments including Illumina HiSeq 2500, all the way through the NovaSeq 6000 platform, using both routine 2x150 paired end chemistry, as well as a novel 2x250 paired end chemistry. And we'll get into the impact of longer reads in a bit. We also were able to get some access to some machines from the MGISeq group, their BGISeq 500 and MGISeq 2000. Not as many replicates, but we had at least one for each of the 3 cell lines. And we also pulled data from our own sequencing as well as some publicly available Genome in a Bottle data on the NCBI FTP for long read platforms such as the Oxford Nanopore. We used our own PromethION, as well as some publicly available PromethION data. PacBio CCS, circular consensus sequencing, or hi-fi reads. These all came from public repositories. And we had an early access look at the Genapsys sequencer. We just had one whole genome replicate for the Sun cell line, but it was still an interesting look to compare against the other platforms. We also ran a targeted exomic analysis using LifeTech Ion Proton INS5 instruments. And for each of these, we had multiple inter and intra-lab replicates. The bacterial genomes on the right here come from ATCC reference material MSA 3001. And that refers to not just a set of individual bacterial strains, including E. coli, Staph epidermidis, P. fluorescens, but also a mixed metagenomic sample that contains 10 known individual bacterial strains. And the idea is how well could we disentangle a complex sample and reconstruct the taxonomic makeup of that sample, as well as how well could we sequence and characterize individual bacterial strains that span a range of GC content, ecological niche, extremophiles, halophilic samples with gram-positive or negative staining, and so on and so forth. And here again, we have Illumina platforms, LifeTech platforms, MinION R 9.4 flow cells, as well as an early access to a Flongle low throughput nanopore flow cell, and again, the Genomes platform. So a really wide range that we, again, have made completely publicly available. And this kind of gets into the sequencing outcomes, the depth and mapping efficiencies. Within the human samples, we broke this down into all the short read platforms. You can see we have an array of mean genomic depth for every sample. We also have some what I call shallow or low throughput runs, again, on the Genomes, the Flongle, and the MinION flow cell. We have some long read samples and then the exomic samples. And you can see here the mapping efficiencies along this row broken down by reads that were uniquely and primarily mapped, reads that were mapped to multiple places in the genome, reads that were marked as either PCR, although these were all PCR-free libraries, so optical duplicate reads were marked, and reads that, when completely unmapped, couldn't be placed anywhere in a reference-based alignment. And you can see there are some pretty platform-specific patterns. Illumina, their high-seqs fall in a pretty tight range. The NovaSeq is a little bit better. The exomic data, it makes sense that it's a greater proportion is uniquely mapped because we're targeting so-called easier regions of the genome. And we'll talk about difficult regions for sure throughout the call. And you can see long reads, especially PacBio CCS, are the most, give you the greatest mapping efficiency. And this kind of recapitulates itself throughout the rest of these panels. You see where unique mapping is down, multi-mapping is up. Duplicate rates are pretty platform specific, and unmapped rates are as well. We don't show some of the PromethION samples here because they have quite low mapping in some cases, because even though you're sequencing very deeply, there are some very short reads that don't align and bring down the overall mapping percentage. Same idea here for the bacterial data. We have a really wide range of sequencing depths, depending on if we're sequencing an individual strain or that mixed metagenomic pool. And here, again, the same analysis shows us a platform-specific pattern, but also a sample-specific pattern. So you can actually see staph epidermidis was not as efficiently mapped as some of these other taxa, reflecting that the quality of your reference genome is going to impact your ability to map, which is pretty intuitive. Focusing now on just the human samples, putting the bacterial data on a shelf, we wanted to look at all these different metrics to compare and contrast the platforms. And the very first thing after genome alignment was to look for coverage. And here, somewhat dramatically, we refer to the classes of evil, the regions of the genome that are most difficult to characterize, most difficult to perform a read-based, reference-based read alignment. And these come from the UCSC repeat masker tracks that are available on their table browser resource on their website. It should be noted, we had a wide range of sequencing depths, but in this case, we normalized all our data. We downsampled everything to a mean genomic coverage of 25x. So about 25 reads on average covering any one particular coordinate in the genome. And you can see the coverage profiles are quite tight. There are some statistically significantly greater distributions of coverage in some platforms than others, depending on the context. But what we thought was really interesting was to look at a kind of instrument by instrument performance against every other instrument. So for simple repeats, for satellite, centromeric, telomeric regions, for long tandem repeats, all of these classes of evil, if you compare a given platform against every other platform, how does it perform? So you can see HiSeq 2500 was unfortunately kind of the loser, so to speak. We don't want to call anyone a winner or a loser, but covered simple repeats at a lower rate than any other platform. In contrast, our long read data, PacBio circular consensus sequence or PromethION long reads were able to cover these regions greater than other platforms. And you can see that throughout all these categories with a mixed signal in between for other platforms. The HiSeq 2500 and the BGI-seq 500 had the shortest read chemistries at 2x125 and 2x100 respectively. So that brought down our mapping rate. So that gives a sense of how these platforms perform in terms of capturing the genome. In terms of accurately sequencing the genome, we looked then at mismatch rates. So once you generate a BAM file, you can actually interrogate it to pull out error rates or measurements of where a read nucleotide disagrees with the reference. And again, we stratified this by all these different classes of evil, and we see very platform and context-specific mismatch rates. So generally, although our long-read data covered these regions better, the error rate in nanopore is still a little bit, it's getting better all the time, but it's still a little bit below the bar set by short-read data and PacBio hi-fi data. You could see the relative mismatch rate for each of these platforms here. We also looked more specifically at homopolymers and short tandem repeats. So homopolymers are stretches of a single nucleotide that are repeated over and over again for a given length. And we see that as that homopolymer grows, we get an increase in error rate for all of the short read platforms. In brown here, you see the PacBio CCS data starts off pretty strong, but by the 75 length homopolymer fits in more or less with the short read platforms, whereas PacBio was more flat throughout, irrespective of the homopolymer length, but stayed relatively high compared to the others. Here we're looking at STRs, and entropy is a measurement of complexity of a given region. So the lower the entropy, the more repetitive or simple the stretch, and an entropy of 0 is essentially equivalent to a homopolymer. And you can see expected similar patterns here. Okay, so that's kind of a naive agnostic approach to look at mismatch or error rate, but can we actually leverage what we know about these cell lines to characterize expected variation? And this is where the genome and bottle resources that I mentioned earlier tie in really nicely. Some work from that group, that consortium, provides high confidence variant call sets. So regions of each of these cell line genomes where you can examine known variants that are based on a multidimensional approach, sequencing very deeply for many different platforms and creating a consensus set of high confidence calls. And there's a paper that is a preprint that came out in December detailing the version 4.2, the latest version that is now available on that GitHub and the FTP that I showed earlier. So we wanted to leverage this because it's a really powerful tool to characterize our sequencing and variant detection ability in these cell lines for each of our platforms. And the first thing we wanted to do was say, well, there are a lot of different algorithms out there for calling germline variants. Which one seems to be performing the best? Which one is giving us the best bang for our buck? And so we used 4 commonly used softwares, Deep Variant, which is a machine learning based variant caller, GATK, which is quite commonly used, and Sentient, which is an emulator of GATK. It's now commercially available. It in theory gives you exactly the same results as GATK, but a little bit, not a little bit, actually quite faster, up to 10 to 100 times faster than the GATK processor, and then Strelka 2. But what we see is that by measuring the level of sensitivity and precision based on these benchmark variants, Deep Variant seems to perform the best. We get the highest sensitivity, the highest specificity, although it is worth noting that that machine learning variant caller was trained specifically on these EBV immortalized B lymphoblastoid cell lines. So there may be a little bit of over-parameterization, overfitting. That's something to keep in mind. Otherwise, we would say that Strelka 2 is as precise, GATK is as sensitive, and Sentient is not far behind either and provides really nice speed up. The other thing that is really noteworthy these days, especially for anyone who's been in this field and has seen the relatively, the performance of long read variant calling hasn't quite been up to the level of short read accuracy, but that's changing. So certainly in the CCS data, we see levels that are up there with any of the short read platforms. And even using CLAIR2 to call variants within nanopore data, we still get pretty good levels, about 0.8 to 0.85 fraction sensitivity and over 0.9 precision. So we're starting to get into the respectable range, the range where you can pretty reliably use nanopore to call variants. And that's actually very exciting and has really big ramifications for the future of the field. Again, though, this was looking kind of at the genome-wide snapshot. We wanted to bring back these classes of evil, and these are going to come back throughout this talk, in the hardest, most difficult regions of the genome to characterize. How are the variant colors performing? Here, we just focused on deep variant because it gave us the best performance. And you can see that depending on where in the genome you're looking, you're going to actually have lowered rates of detected accurate variants. You could see, especially in regions that are defined as being low complexity or simple repeats, we start to bring down those levels. And what's also interesting is that in regions like satellite centromeres and so on, we see a genome-specific signal. So it seems like these circles, which represent the Ashkenazi mother, we can't quite reach the level of sensitivity, for the most part, that we can for the father and the son. And that just reflects a little bit about maybe there's room still to improve the benchmark. And it's an ever-improving resource. We're on version 4.2 now, but it's certainly not set in stone. So this is something that to keep in mind as you look carefully, depending on where in the genome you are. We also, of course, wanted to integrate the LifeTech exomic data, so we focused our analysis just to the exome regions, and we see, again, pretty good performance. What's really interesting is that even using the native TVC suite or torrent variant caller for the Proton S5 data, we still get relatively weaker performance compared to Illumina and BGI samples. And even the nanopore in dark green here seems to fit in pretty well with the LifeTech data. So that maybe says something about the improvement of some of these instruments. We also wanted to provide the same analysis in medically relevant genes. So using the ClinVar database from NCBI or the OMIME, the Online Mendelian Inheritance in Man database, we see, again, the same kind of performance. BGI and NovaSeq 2x150 and 2x250 especially give the greatest level of sensitivity and specificity, with the Illumina platforms not far behind. And again, Nanopore is getting there, and PacBio is already there using hi-fi reads. One other way to approach this was to actually look at the intersection of known true positive calls. So again, pulling from the Genome in a Bottle benchmark, how many of these known true positives in high confidence regions are we calling? And we created what we call Altoona plots, named after the person who created these plots. He will forever be remembered for making Altoona plots. For each of these platforms within SNP and INDEL regions, how many of these true positives are we detecting versus how many are missing in blue? And you can see a very platform specific pattern. HiSeq 2500 missing the most SNPs. And then long reads, Nanopore and PacBio, Nanopore especially failing to detect some of the known high confidence indels or insertion deletion events within these regions. This is a bit of an overwhelming slide, but we love it and we wanted to include it. This is a heat map or a matrix looking at all of the variant allele frequencies, or VAF, that we kind of joke internally looks like a bath robe that you might wear. So we call it the VAF robe, soon to be trademarked and available for, no, it won't be available for purchase. But the interesting thing to note here that recapitulates what we saw before is that where each sample for each platform is a row, we see that long read data in green here cluster separately from all the short read data, because in blue, there are just all these missing true positive calls. Whereas in each of these other clusters, we see, as you would expect, stratification by cell line, the son, the mother, and the father here. And we can actually see the distribution of homozygous variant alleles in red and heterozygous variant alleles in orange. And what's really great about this is a family trio. So we can actually look at the variants called at a given site for each of the three cell lines and try to determine, are we seeing Mendelian violations, allele combinations that shouldn't be possible given the family trio nature of these samples. Fortunately, although we do tend to see some Mendelian violations, they're quite minimal and they really just seem to be technical noise. So when we look at our known true positives and we use a software called VBT to detect Mendelian violations or Mendelian misinheritance, we see just platform-specific the violations popping up, although you can see it's about 15,000 out of the hundreds of thousands or millions of variants that we're calling. So this is an extremely low rate that is associated with technical noise. So that's a nice affirmation of the accuracy of the variant callers that we're using. And we also finally, within these sort of shorter variant events, we wanted to look at how well can you characterize indels based on the size of that insertion or deletion. And what we see, as we saw before, is that although with nanopore permethyon data, we still are capturing many of these indels, as we get longer and longer insertions, the nanopore curve kind of falls off, whereas the NovaSeq 2x250, PacBio Hi-Fi, and even some of the HiSeq and BGI samples are able to detect these indel events up to 50 bases in size. That's sort of where SNV calling cuts off at 50 base insertions or 50 base deletions. What's also interesting is that we see more separation for insertions between platforms than we do for deletions. We weren't really quite sure why this was. It was a little beyond the scope of the study, but it's something that might be interesting to characterize further as a follow-up work. The other and last sort of set of analyses we wanted to do in variant space was to look at structural variants. So characterizing larger structural chromosome level events, transversions, translocations, large insertions or deletions. There have been several studies that have characterized these in these cell lines and other genome and bottle reference material cell lines. But what we wanted to do was to provide an intra-platform comparison. And what's noteworthy is that although there is a reference set of SVs for HD19, our study was rooted in HD38, the latest build of the human reference genome. And so there isn't per se kind of reference material standard for SVs yet. But what we were able to do was to lift over the HD19 coordinates to HD38 as best as we could and try to benchmark against those. Not only did we try to benchmark, but we also tried to get at the question of what's contributing the most to the difference in signal that we're seeing in SVs, because SV calling is quite noisy and can differ not only between sequencing instruments, but even between replicates on a particular instrument. So what we did was we wanted to look at the impact of SV callers, the impact of sequencing platform, and the impact of different libraries being sequenced on a platform. So here, the impact of callers, what we did was we merged all of our outputs across a given platform per sample per caller. We looked at the Ashkenazi sun. We merged across all the different laboratories that perform sequencing. And then we looked at the callers themselves. In this case, Manta, Lumpy, and Deli are the three kind of most commonly used SV callers these days that are then meta-analyzed by this package called Survivor that can retain an SV if it's being called by multiple callers. And what's the kind of big takeaway here is that we're seeing, first of all, a lot of SVs that are missed, and that's just a product of not only depth of sequencing, but being able to call across multiple callers. So some of these callers are dropping out and therefore reducing our true positive rate. But we also see a lot of caller specific things that may be false positives, or there may be something happening with the criteria of inclusion for each caller. Finally, the other thing worth noting is that within these cell lines, we see a lot of large scale deletions. You can see here the 100 base to 1 KB range. We see a lot of these structural events are deletions. We also see smaller translocations, but we tend to leave those out. There's some good literature as to why those are typically considered to be false positives. And I'll kind of leave it at that because that's a whole can of worms. We also wanted to look at the impact of platform, as I mentioned. And what's interesting here is that we still see some platform specific differentiation, but we also see a lot of harmony between the platforms. And in this case, we can only look at some of the high-seqs because for the other platforms, as I mentioned before, we only had a few replicates, not enough for a replicate-based comparison. And the same kind of pattern of deletions, duplications, inversions, and so on are seen. So it doesn't really matter how you filter, you're going to see the same genomic distribution, which is a nice confirmation of what we're seeing. So to get at the overall trends for SVs, we provide readers with an expected distribution of structural variants, what they might see depending on the SV caller that they use, what they might see depending on which platform they use. And I didn't show this in great detail, but concordance between replicates is the highest out of each of these filtration approaches that we took. And lastly, we also did one last validation. Are we seeing spurious SV calls that are occurring over and over again in some genomic region that might be an area of low complexity or something that's causing our SV callers to kind of be like moths to the flame and call many, many false positives? But we're not really seeing that to be the case. There are, if we look at 100 KB windows, almost all of those windows contain just one SV. So we're not getting some window in the genome with dozens of SVs that might raise an eyebrow. They're pretty well distributed throughout the genome, which is, again, a pretty nice affirmation. Okay, so that's just to shift gears here. Those are more or less the major analyses we ran on the human samples. Now I want to jump back to those bacterial reference materials that I showed previously. You may remember we sequenced individual bacterial strains as well as a mixed metagenomic pool of 10 bacterial species and wanted to see how well we could reconstruct those. The individual strains looked more or less as you would expect, not a lot to show there. But what we found was really interesting was the inconsistency of taxonomic distribution based on which platform you were using. So each of these instruments, when we characterized the reads they produced with a taxonomic read classifier, we saw a really wide range of percentages for each of the 10 genomes that were included, each of the 10 bacterial strains that were included in the mixed metagenomic sample. And you can see from the Illumina-MiSeq, Genapsys, LifeTech, S5, and PGM, as well as the two nanopore platforms, we see a really platform-specific distribution of these genomes. And what you can also see is that these edge genomes seem to be much less, or they seem to be underrepresented relative to the middle ones. Keep that in mind. I'll show you in a moment why that is. But just as another way of looking at these data, if we look at the normalized coverage, the number of reads for a given sample, and where those reads fall along the spectrum of GC percentage, you could see, first of all, very, very specific profiles for a given genome. Staph epidermidis, or STE, has a relatively AT-rich genome. So you could see about 15 to 30% GC is predominantly what we see. In E. coli, it's more middling, and in Pseudomonas fluorescens, it's more GC-rich. You could see across the different platforms, we don't have any kind of clustering. We don't have platform-specific or sample-specific clustering for the individual strains. But for the mixed metagenomic strain, we see way more of a distribution, and we even see some clustering by platform, which is interesting. We also show that the level of entropy per sample is much higher in the pool than in the individual strains, which makes sense. It's to be expected, but it's something that should be considered when you're sequencing a more complex sample. And as I mentioned before, low and high GC taxa seem to be underrepresented in these data. So particularly Staph epidermidis, I mentioned it's an AT-rich genome. That tends 28% GC content. That tends to be underrepresented. So we have 10 genomes included here. We expect about 10% coverage per genome, but in this AT-rich genome, And in this GC-rich M. luteus genome, we see significant underrepresentation. We also have some differentiation depending on gram-positive or gram-negative, and that kind of gets into the ecology and salinity and cell wall characteristics of each of these taxa, something to be kept in mind depending on how well you're able to know your sample set a priori. And again, the winners here, so to speak, are the taxa with middling GC contents. Those seem to be captured the most easily. And we ran some permutation tests and found over and over again that these genomes with more middling GC content are going to be overrepresented in these samples, which was really quite interesting. So one last note, as I mentioned up top, we believe very strongly in reproducibility, in open science, and in sharing as much as possible. So again, all of these data have been deposited to SRA, and we even have a Wasabi S3 endpoint. If anyone wants to work with us and pull some of these data, we're happy to share. And we're also happy to share all the code. So this is just a snapshot from a GitHub repository that we generated a couple weeks ago before submitting our revision, containing all of the bash scripts, all of the Slurm scripts to submit jobs to a scheduler, any kind of novel Python or R or bash scripts that we used for this data, as well as our visualization notebooks for generating figures. And so that's all freely available. And with that, I want to thank you guys for your attention. I appreciate y'all being on this virtual webinar. Hopefully we can return to in-person webinars and chatting over drinks afterwards, but for now, this will still have to do. And I want to thank members of Dr. Mason's lab who were integral in this work, some other folks throughout the community, and then again, the NGS2 working group, without whom none of this could have been possible. So once again, thank you. And I'll be happy to take any questions and pass it back to Dr. Jones.
Jonathan, thank you. That was very interesting. I know I have a number of questions, let me, and don't take away your slides, because I want to
Sure.
ask a question related to slide. So related to the metagenomic part, maybe the part that you just went over, if you can go back to the GC content relationship between the different pool, especially this one. So, sometimes I know in metagenomic context, there may be a need to potentially quantify in a metagenomic context, how frequent does one particular type of organism appear versus another. So I guess the question is, it seems like that this curve that you have is really strongly related to GC content. And I was just wondering, do you feel like it's strong enough where you could, in essence, if you were trying to quantify how much is present, could you compensate if you knew the GC content of the particular organism to, say, okay, if it's high or low in GC, it'll be underrepresented. Therefore, I might be able to compensate in terms of estimation. And the same thing if it's in the middle, in one sense, depending upon what else is present, and that's where kind of the math gets interesting, because in one sense, it kind of depends on what else is present and whether it's over or underrepresented. But do you feel like you could somewhat compensate for that and not mistake let's say a 30% presence for a 15 or 10% presence, or is that maybe reading too much into the data?
Yeah, I think that's an excellent question. And what I would say in the first place is that, this study is relatively underpowered to definitively answer some of those questions. So the hope is that this could be a launching point for further research into this kind of question. But one thing as a caveat that I would note that we didn't include in this talk is considering the actual genome size of these taxa. And there is some difference to be said in terms of the actual genome content, right? If we included a human sample here, it would probably be overwhelmingly represented because the human genome is so much greater and we could contribute so many more nucleotides to our overall pool. But at the same time, I do think there is something to be said for what you were getting at in terms of accounting for or normalizing for differences in GC content and genome size. And that could be something that could be modeled and perhaps incorporated into metagenomic classifiers, because the way we ran it, we kind of used the naive approach of take all the reads, use a k-mer hashing approach and try to assign a taxonomic assignment to each mate of, because this paradigm sequencing, to each mate. If they matched, then we gave that read pair that taxonomic assignment. And there could be, I think, something a little more sophisticated to be done to compensate for the potential makeup of your sample. We had the benefit of knowing exactly what was in this because this is a well-characterized, commonly used, I think it's often used for spike in, for quality control. So we knew what was in the sample ahead of time. For, an environmental sample where you don't really know what you're going to get, that might be trickier, but it's something that hopefully this work could spur and help to create in some future work or some future software.
Okay, well, thanks. And so I wanted to mention Chris Mason's comment about the emphasis on the classes of evil related to different repeat regions or complex or low complexity regions of the genome. I want to talk about classes of evil variants to detect. So, so you had a very interesting slide on the structural variants, which to me would be, of all the variants, maybe the evil variant detection, because I think it showed that structural variants are really, really, really hard to detect, at least what I see coming out of some of your work. And what I was wondering is, I was wondering if we can make an analogy to, let's say about 10 years ago, there was a similar kind of interest in, for example, detecting fusions events, for example, in RNA, and how initially a lot of the original fusion callers, if you compared them, they would be all over map, even for the same data. And you would get low reproducibility between callers, often low sensitivity. But now we've seen over time, a lot of fusion callers have matured and you're getting better reproducibility between callers, you're getting better sensitivity to fusion events and things of that nature. And I'm wondering if you feel like for structural variants, what is the path forward? For the fusion events, it seemed like we were able to, cure it with better methods on the bioinformatics side. I'm wondering on the structural variant side, is this a bioinformatic problem? Is it a read depth or read length problem? Is it a platform, problem? Is it all of the above? And what is needed to sort of move to move our yardstick in terms of what is acceptable structural variant performance and what we should expect going forward?
Yeah, again, great questions. It's interesting you mentioned gene fusions in particular, because one of the other SCQC2 subgroups is actually looking specifically at reproducibility of gene fusion events from RNA-seq data. And that's a study that we'll be wrapping up. I don't know if either Dr. Boku or Dr. Zhu are on the call, but we're kind of trying to wrap that up as much as possible. And exactly to your point, we're finding a lot more consistency in these calls than we might have expected, although still some inconsistency that we're trying to characterize. And what has helped, and I think this is kind of the answer to your question, is the introduction of long reads, particularly long reads that have significantly improved accuracy compared to where the state of the art was 5-10 years ago. We show in this paper to an extent, and there's some good work from other SV algorithm announcement papers, that the implementation of PacBio CCS reads, of higher fidelity R9.4 and R10 nanopore reads tends to give us a wider snapshot and a more reproducible snapshot of structural variants. That said, it's still early days, as you mentioned, and I think, you asked, is it a bioinformatic problem or is it a platform problem? And I think it's still a little bit of both. And the idea, hopefully, would be in tandem as sequencing instrument technologies improve, our computational abilities improve. And I think some of that will come with approaches like what Survivor does, which I mentioned, which tries to aggregate the union or the intersection of SV calls and harmonize them and give you as best an estimate as it can as to what may be true and recapitulated and echoed by multiple lines of evidence versus what might be a false positive or what might just be the product of one computational approach.
Okay. And I guess my last question, if you go back to one of your early slides, you were looking at the mismatch rate of different platforms relative to, yeah, here we go. And you mentioned the PacBio and using the circular consensus sequencing. And, you know, it performs, in one sense, related to these classes of evil regions, if you want. It's better, or at least comparable to many others in almost every instance. And the question I had was, in one sense, you know, since PacBio has a little bit of flexibility, or at least, you know, my understanding, there's a little bit of flexibility related to the circular consensus in that you can prescribe how long, you know, your movies, you know, can last, and therefore it relates to, you know, the size of the insert, if you like, and how long it goes. I guess I was wondering, was there any design related to how you engineered the PacBio to get circular consensus, you know, quality values that would be this, or was this just like the normal operation? So I guess what I'm wondering is, with the others, you know, the quality is especially for like Illumina, I mean, it is what it is, and there's not much flexibility in terms of, say, you know, taking a parameter and adjusting something to improve base quality. The circular consensus, it seems like maybe there's a little bit more flexibility. And I didn't know if you did any studying or engineering or whatever to say, well, yeah, we targeted for a mismatch rate here and we got that. And what maybe that flexibility is, you know, for PacBio versus some of the others.
Yeah, that's such a great question. It's something that I think could be its own follow-up study. The CCS data, that was one of the only data types that we didn't generate for the study. We pulled
Oh, that's right. I
exclusively.
forgot.
Yeah,
that's right.
So the NCBI-FTP they do provide sub-reads, I think, for hi-fi reads, but we just use the FASTQs that they generated and recommended. But, you know, I guess one thing that is worth noting is variant calling, epigenetic modification calling from PacBio, CCS reads, is becoming more and more tractable as it's becoming cheaper and easier to loop around a given fragment over and over again. I am now curious about the relationship of sequencing of nucleotide fidelity to the number of subreads that you need. And I think there have been some interesting talks at recent conferences announcing, you know, PacBio Hi-Fi can routinely reach Q40, Q50 based on the number of, sorry, my cat is flying all over my desk if you saw me shake like I had an earthquake just now. But as that technology improves, I think it becomes easier to incorporate more and more subreads. And therefore, I mean, we see in these data, even with hi-fi reads from 2-3 years ago, the level of improved fidelity. So that's actually really interesting. I wonder to what extent we could pull out even more raw data and run subread titration experiments and see how that impacts our ability to call variants and even structural variants.
Yeah, I mean, there's definitely a cost to that, because, you know, the longer you let the movies run, you know, you're basically taking up more time on the instrument, and, you know, not every, you know, there's still variability in the length. So even though the movies are running along, you may get, you know, much more subreads for some versus others. I mean, there's a whole complex question there, but I'd forgotten. That's right. You said you had pulled this data from another source, and I didn't. I know in the Weill Cornell lab, I know you work a lot with PacBio, so that's why I was wondering.
We do have one, yeah.
If you had done any engineering there. So okay, all right. So I think that's all the questions that we have right now. Well, Jonathan, thank you so much for your presentation. I know I have learned a lot, especially about some of the new platforms and seeing the progress, you know, of the Oxford Nanopore and how they're working. And really, in one sense, it's really great to see, you know, the agreements that you're seeing with the different platforms in this context, at least in the human genome. I know there's some other challenges that are, you know, when you have GC variation in these specialized, you know, kind of genomes. But I'm just very happy that there's, you know, there's good consensus on several fronts related to this variety of platforms. I think it speaks to the ability for us to reproduce each other's work, even though we may be using different platforms, to see, you know, the agreements, you know, with some of these major platforms here. So very, very optimistic, you know, from that view. In one sense, we've come a long way from, you know, I'd say the mid 2000s when we were, you know, looking at 50 to 100 base reads, and we were getting poor quality at the end of some of them, and we've come a long way in just a relatively short period of time. Oh, actually, we have a question who just came in. Hang on. So Good Haryani is asking, is there a theory as to why the 2500 missed a lot of the true positives in the NIST sample? And I don't recall that in particular.
Oh yeah, that's a good question. I can pull that up. I do have one overarching observation about all of these, and that is the read length has an outsourced impact on our ability to detect some of these true positives. So I think the question is, sorry, can you still see this?
Yes, yes.
Great. The question is, why are we missing so many of these true positives in HiSeq 2500 and in these like ALUs, L1s, L2s? And that was one of the only platforms where we did 2x125 sequencing. So our insert size was shorter, our read length mates were shorter, and that may have contributed to our inability to detect some of these true positives.
Yeah, and that makes sense. And especially on the INDEL side, you see the the huge impact, and especially for the 2500. And yeah, that I can certainly understand that. That's an interesting observation. So in one sense, size does matter, right?
It seems that way.
It seems that way. Okay. Well, Jonathan, again, thank you very much for your presentation. Really enjoyed it. Looking forward to hearing more about that and reading it in the manuscript when it comes out.